Okay, so picture this for a second. You're just on a rock floating in space.
Mm-hmm.
And this isn't some calm, peaceful rock, right? Billions of years ago, this thing started as like a giant ball of flaming molten stuff.
Yeah, constantly getting absolutely pummeled.
Totally. Asteroids, comets, just bam, bam, bam.
And the water.
And here's one of those amazing details. Yeah, the water. Those incoming rocks and ice balls, they likely delivered it.
Incredible when you think about it.
So this fiery planet slowly starts to cool down, and eventually it rained.
For ages, apparently.
Yeah, just rained and rained, flooded everything.
But down at the bottom,
Yeah.
something interesting was happening.
Exactly. Down in those early oceans, especially near things like hydrothermal vents, spewing hot mineral-rich
A
chemical.
real chemical soup.
Something truly extraordinary began to stir because somehow out of that violent mix we eventually got, well, us.
Yeah, which is quite the journey.
Welcome to a deep dive into biology.
Yeah. Exactly. And our journey today, it's really inspired by this fantastic source material you share, that YouTube transcript. It does such a great job breaking down complex ideas.
It really does.
So our mission really is to go through it together, pull out the absolute core essentials of biology, find those, surprising connections.
There's those aha moments.
Yeah, nail down those aha moments that turn something as vast as life into something understandable and genuinely exciting.
That's the goal. We're going to tackle the really big questions here. Like, what is life fundamentally? How does it work down at the tiniest level of the cell? Where does it get the energy to do everything?
Crucial point, energy.
How do traits get passed from one generation to the next? And even how can tiny accidental changes
And
mutations lead to massive shifts over time? Evolution, diseases, all that.
And right off the bat, the source hits you with this powerful idea. At its core, biology is really just chemistry in disguise.
I love that framing.
Yeah, when you strip away all the complexity, you and I are, in a sense, just a big ball of molecules that can make funny sounds.
Which, okay, brings us right to those fundamental building blocks, the key molecules making up life. The source highlights 4 big ones. First, carbohydrates, the quick energy guys.
Sugars and starches.
Then lipids, they're for storing energy long term, but also crucial for building structures like cell membranes.
Fats, oils, yeah, very important.
Proteins. These seem like the absolute workhorses. They make up our tissues, but they also do incredible tasks like being enzymes.
Definitely the workhorses.
And finally, nucleic acids, the family DNA belongs to.
Enzymes. This is where the chemistry really comes alive, like you said.
Yep.
The source emphasizes these are special protein catalysts. What that means is they speed up specific chemical reactions by just staggering amounts.
So they make things happen faster.
Way, way faster. They can either build larger molecules or break down complex ones. Life simply couldn't happen fast enough without them.
Wow.
Think about the example given, lactase. It's the enzyme that specifically breaks down lactose, the sugar in milk.
Okay, I know people who have trouble with that.
Exactly. Without functional lactase, digesting milk is incredibly difficult. It just shows you how critical these single molecules are for, well, everyday function.
So we've got the basic chemistry humming along thanks to enzymes, but it still doesn't quite get to that big question. What is life?
Yeah, the philosophical bit.
I love that the source acknowledges scientists still debate this, but, we can definitely tell the difference between, say, a cat and a rock based on the characteristics it lists.
There are some clear signs.
The cat does things, right? It takes in food, gets energy metabolism, it grows, changes development, it can make more cats reproduction, and it reacts to its environment response.
Well, the rock.
The rock just sits there. It doesn't really do any of that.
Right. And if you zoom in on that cat or a plant or even just a tiny organism in pond water, what's the common denominator? Cells. The source is very clear. Every single living thing on Earth is built from cells. It's the fundamental unit.
Okay, and these cells, they come in 2 main flavors, as the source describes them. You've got prokaryotes.
The simpler ones?
Right, simpler, always single-celled things like bacteria and archaea. Their inside structure is pretty minimal. They don't have those membrane-bound compartments, so their DNA is just kind of floating around.
Chilling in the cytoplasm, yeah.
Then you have the eukaryotes. These can be single-celled too, like some protists.
Right, like amoebas.
But they're the ones that can build big, complex organisms. Fungi, plants, all animals.
Ah.
Us.
And the key difference is that compartmentalization. Eukaryotes have these fancy membrane-bound organelles.
Like a nucleus.
Exactly, like a nucleus that carefully stores the DNA, keeping it separate, and mitochondria for energy and so on.
Okay.
And speaking of organizing things, with just the sheer diversity of life built from these cells, we obviously need a system to keep track.
Right, you can't just wing it.
No. The source touches on how we classify life using taxonomic ranks.
Yeah.
Kingdom, phylum, class, order, all the way down to species.
That Linnaean system.
Yeah. It points out that just calling things cat, dangerous cat, water cat, it's not very helpful scientifically.
No, probably not.
So we give every unique species a two-part scientific name, the genus and species. Felis catus for the house cat. Avoids confusion.
Makes sense. Okay, but whether you're a simple prokaryote or a complex eukaryote, there's this universal challenge, staying stable inside, right? The source introduces homeostasis.
Absolutely crucial concept. Homeostasis.
It's essentially about keeping certain internal conditions in check, so yeah, don't die. A bit blunt, but yeah.
It's accurate. And you see homeostasis in action in your own body constantly. Like when you get too hot,
sweat.
you sweat to cool down.
Yeah.
When you're cold,
Shiver.
shiver to generate heat. Cells do this too, but more on a chemical level, balancing concentrations. The source specifically mentions keeping the pH level stable.
Okay, why is pH so important for a cell?
Because those enzymes we talked about,
The catalysts? Yeah.
they are incredibly sensitive. They only work properly within a very narrow range of conditions: temperature, pH, salt concentration. If the environment changes too much, the enzyme can denature.
Denature. What's that?
It basically unravels, loses its specific shape. And its shape is essential for its function. So if it denatures, it stops working.
Okay, so maintaining homeostasis is literally life or death for a cell because it keeps the enzymes working.
Exactly. Keeps the machinery running smoothly.
And how does the cell manage that? How does it control its internal environment so precisely?
The source points right to the cell membrane, the gatekeeper.
That semi-permeable phospholipid bilayer. Sounds fancy.
It does, but it's just two layers made of these funky-looking molecules, as the source calls them, phospholipids.
Right, I remember reading about those. They have a head and two tails.
Yep. One end, the head, loves water, polar. The other end, the tails, hate water, nonpolar. So they naturally arrange themselves in two layers, tails facing inward, away from the water inside and outside the cell.
Creating a barrier.
A really effective barrier, especially to water-soluble things. This structure is brilliant because small molecules that can dissolve in lipids like oxygen and carbon dioxide can just slip right through. Even water can pass relatively easily.
But not everything.
Not everything. Larger molecules or charged particles like ions, sodium ions, potassium ions, they can't just cross that lipid core.
So how do they get in or out if the cell needs them?
They need special protein channels or transporters embedded within the membrane. Think of them like specific doors or gates. And the cell can control whether those doors are open or closed.
Giving it control over what enters or leaves? Very clever.
It's incredibly sophisticated control.
So how do things actually move across this membrane then? The source covers a few ways. First, diffusion.
Diffusion, the simplest. It's just particles moving passively, automatically, from where there's a lot of them, high concentration, to where there are fewer, low concentration.
Like a smell spreading across a room.
Exactly like that.
Yeah.
No energy needed. It just happens down the concentration gradient.
Okay. What about water? You said it moves easily, but the source mentioned something specific.
Right. Water movement, osmosis, is a bit special. Water tends to move towards areas with a higher concentration of dissolved stuff solutes.
Higher concentration of stuff.
Okay, this is where that salt water example from the source is so powerful. If you drink a lot of pure salt water,
Which you shouldn't do.
definitely don't.
Yeah.
There's suddenly a much higher concentration of salt outside your cells than inside. So water actually gets pulled out of your cells by osmosis, trying to dilute the outside saltiness.
And that dehydrates your cells. Bad news.
Very bad news. Shows how important water balance is.
Okay, so diffusion is passive. What if a cell needs to move something against the flow? Like from low concentration to high?
Now that takes effort. This is called active transport.
Active meaning it needs energy.
It absolutely requires energy. You're essentially pumping things uphill against their natural tendency to diffuse.
And where does the cell get this energy? For active transport, for everything else?
Universal energy currency for life. Adenosine triphosphate ATP.
APP, I've heard of that.
The source calls it kind of important, which is a massive understatement.
Right.
Energy is released when a chemical bond between the last two phosphate groups in the ATP molecule is broken. It's like a tiny rechargeable battery. Every single organism, every single cell relies on ATP.
Okay, so if ATP is so vital, how do you make it? Where does it come from?
In most complex cells, the main factory is the mitochondria, through a process called cellular respiration.
Cellular respiration, okay.
The source gives the basic recipe. You take glucose, that's a sugar, and oxygen.
Stuff we breathe in and eat.
Exactly, and you react them together. The outputs are water, carbon dioxide.
What we breathe out.
Right. And crucially, lots and lots of ATP energy. That's the main payoff.
Glucose plus oxygen minus water plus carbon dioxide plus ATP. Got it.
But where does that glucose come from in the first place? This is where organisms differ quite a bit.
Okay.
Heterotrophs, like us humans, are the eaters. We get glucose by consuming food plants, animals, and breaking down the sugars or complex carbohydrates in it.
We find our glucose.
We find it, yeah. Autotrophs, like plants, they make their own. The source humorously says they make their own glucose by staring at the sun.
Ah, photosynthesis, right?
Photosynthesis, exactly. This happens in special organelles called chloroplasts. They contain chlorophyll.
The green pigment.
That's the one. Chlorophyll is amazing at absorbing red and blue light, but it reflects green light, which is why most plants look green to us.
Okay.
Plants use that captured light energy to split water molecules and take carbon dioxide from the air. And through a complex series of reactions, they convert it into glucose. And as a byproduct, they release oxygen.
The oxygen we need for our cellular respiration, it all connects.
It's a beautiful cycle.
Okay, let me recap the energy part. Whether you ate something to get glucose or you're a plant that made its own glucose via photosynthesis, once you have that glucose, you can feed it into cellular respiration, combine it with oxygen, and generate that vital ATP energy.
You got it. That's the core energy flow.
Ooh.
Now, it's interesting. ATP is actually a nucleotide.
Oh, like in DNA.
Exactly. And the most famous nucleotide-based molecule is, of course, deoxyribonucleic acid, DNA.
The blueprint of life?
The blueprint. The source describes its iconic double helix structure.
Yeah.
2 strands, like a twisted ladder. The sides of the ladder are made of a sugar phosphate backbone.
Okay.
But the really important part, the actually important part, as the source puts it, are the rungs of the ladder. These are made of pairs of nitrogenous bases.
A, T, C, N, G.
Adenine, thymine, cytosine, and guanine. And they have specific pairing rules. A always pairs with T, and C always pairs with G.
TCG. Got it.
They're held together across the gap by hydrogen bonds, which are relatively weak, allowing the strands to be separated when needed.
Okay, so how does this structure, this sequence of letters, actually hold information?
Great question. The source explains that a gene is just a specific section of that long DNA sequence, a particular string of A's, T's, C's, and G's.
Like a specific sentence in a huge book.
Exactly. And that sentence acts like a recipe for making a protein.
A recipe for protein. Okay, connecting back.
And circling back, proteins are absolutely critical, remember?
The workhorses.
Yeah. The source emphasizes they do everything. Transport molecules, act as enzymes, form structures, and even determine the way you look.
How does that work? The looks part.
The example of eye color is perfect. It's largely determined by how much melanin pigment you produce.
Okay.
There's a gene called OCA2, which provides the instructions of the recipe for making a protein called P protein. This P protein helps control how much melanin is made.
Ah, so the gene codes for a protein that affects melanin.
Precisely. The specific version, or allele, of the OCA2 gene you have determines the kind of P protein you make, which in turn can influence whether you have a lot of melanin, leading to brown eyes, or very little, leading to blue eyes. It's literally the proteins made from those genetic recipes that shape our physical traits.
That's amazing. But wait, there's a logistical problem, right? In eukaryotic cells, the DNA, the recipe book, it's safe in the nucleus.
Right, locked away.
But the ribosomes, where the proteins are actually built, they're out in the cytoplasm.
Correct.
So how does the recipe information get from the nucleus to the ribosome?
This is where RNA comes in, ribonucleic acid.
DNA's cousin.
DNA's close cousin, yeah. The source notes the key differences. RNA is usually single-stranded, not double. It uses a slightly different sugar, ribose, instead of deoxyribose. And instead of thymine T, it uses uracil, U, So U pairs with A in RNA contexts.
Single-stranded, different sugar, U instead of T. Yeah.
And it's generally less stable than DNA, which is fine because its job is often temporary, carrying messages.
Okay, so RNA carries the message. How does that whole process work, turning the DNA recipe into an actual protein?
It's called protein synthesis, and it involves two main steps, as the source details. First, transcription.
Transcription, like making a copy.
Exactly like making a copy. An enzyme, called RNA polymerase, finds the right gene on the DNA, unwinds that section of the double helix, and builds a complementary RNA strand using the DNA as a template. This RNA copy is specifically called messenger RNA, or mRNA.
Messenger RNA, it makes sense.
The mRNA molecule then peels off the DNA and carries that genetic message out of the nucleus through a nuclear pore to a ribosome waiting in the cytoplasm.
Message delivered. What happens next?
Next comes translation. This is like reading the recipe on the mRNA and actually assembling the protein.
Okay.
The ribosome clamps onto the mRNA and starts reading its sequence in chunks of 3 bases. These 3-base chunks are called codons.
Codons, like 3-letter words.
Perfect analogy. Each codon specifies a particular amino acid. Remember, amino acids are the building blocks of proteins. Now, how do the amino acids get there? Another type of RNA called transfer RNA, or tRNA, acts like a delivery truck. Each tRNA molecule carries a specific amino acid on one end, and on the other end, it has an anticodon, 3 bases that are complementary to a specific mRNA codon.
So the tRNA matches the codon.
Exactly. The ribosome moves along the mRNA, reading codon by codon. The matching tRNA brings the correct amino acid. The ribosome links that amino acid to the growing chain that moves to the next codon and repeats the process.
Adding amino acids one by one.
Until it hits a stop codon on the mRNA. The result is a long chain of amino acids, a polypeptide chain, which then folds up into a specific 3D shape to become a functional protein.
So transcription in the nucleus copies DNA to mRNA. Translation at the ribosome uses mRNA to build a protein.
Wow. It's an incredibly elegant and precise process. Information flow. DNA to RNA to protein. That's often called the central dogma of molecular biology.
Central dogma. Okay. And speaking of the sheer volume of this, genetics is just mind-boggling. You have about 20,000 protein coding genes, right?
Roughly, yeah. But the source points out something fascinating. That's only about 1% of your total DNA.
Only 1% codes for proteins? What's the rest doing?
That's a huge area of research. Some of it regulates gene activity, some has structural roles, some we're still figuring out. It's definitely not all junk. And remember, almost every single cell in your body contains a copy of your entire genetic code. All 3 billion base pairs.
Okay, hang on. This raises that packing question again. The source mentions you have roughly 2 meters of DNA.
2-cell nucleus, yeah.
Packed into the nucleus of a microscopic cell. How on earth does that fit?
It's an amazing feat of biological engineering. It's all about supercoiling and packaging. The DNA double helix isn't just floating around loose.
No.
No, it's tightly wrapped around spool-like proteins called histones. Think of beads on a string.
Okay, DNA wrapped around histones.
These wrapped structures, the DNA plus histones, then coil up further into a fiber called chromatin.
Getting done.
Exactly. And then, when the cell is getting ready to divide, this chromatin gets even more condensed and folded, forming those visible, distinct structures we call chromosomes, often drawn looking like little X's or worms.
Worm-looking things, yeah.
It's like packing a ridiculously long, thin thread into a tiny, tiny box using multiple levels of coiling and folding.
Incredible. And the human genome isn't just one giant DNA molecule, right? It's split up.
Correct. It's organized into 23 different types of chromosomes. And in your body cells, your somatic cells, you generally have 2 copies of each type of chromosome.
One from mom, one from dad.
Precisely. One set inherited from your mother via the egg, one set from your father via the sperm. So you have 23 pairs, or 46 chromosomes total in most cells.
Okay. And for most of these pairs, the 2 chromosomes carry the same genes in the same order.
Yes, for chromosomes 1 through 22, the pairs are homologous, meaning they carry the same genes in the same locations, or loci.
But the versions might be different.
But, as the source points out, the 2 versions of a specific gene on those 2 homologous chromosomes might be slightly different. These different versions are called alleles.
Alleles.
So for most genes, you inherit 2 alleles, one on the chromosome from your mother, one on the chromosome from your father.
And how those 2 alleles interact determines the trait you actually show.
Often, yes. Huh. The source explains the classic concept of dominance and recessiveness.
Right, the uppercase and lowercase letters.
Exactly. If you have one dominant allele, let's use the source's example, B for brown eyes, and one recessive allele lowercase b for blue eyes.
Your genotype is Beeb.
Your genotype is B, and because B is dominant, your phenotype, what you actually express, is brown eyes. The dominant allele masks the effect of the recessive one.
And the recessive trait, blue eyes in this case, only shows up if?
Only if you inherit 2 copies of the recessive allele. So your genotype would have to be a B.
Okay.
The inheritance example in the source is a great way to see this. Imagine 2 parents who both have the B genotype.
So they both have brown eyes.
Right. But they both carry that hidden B allele. When they have children, each parent randomly passes on one of their 2 alleles. So there are 4 possible combinations for the child. BB, 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 or BB.
Okay, 3 of those have at least one B.
Exactly. BB, BB, and B, which is the same as BB, will all result in brown eyes. But there's that one combination, BB.
The 1 in 4 chance.
A 1 in 4 chance, statistically, that the child inherits a B from both parents and ends up with blue eyes, even though neither parent had blue eyes.
Wow. It really shows how your visible traits don't always tell the whole genetic story.
Totally. As the source says, it's what's on the inside that counts, genetically speaking.
Though the source is quick to add that it's not always this simple, right? This neat, dominant recessive thing isn't the only way.
No, genetics gets more complex. There are other patterns. The source mentions intermediate inheritance.
Like blending.
Yeah, where the combination of alleles results in a blended phenotype. Like crossing a red snapdragon flower with a white one can give you pink offspring. Neither allele is fully dominant.
Okay. And codominance.
Codominance is where both alleles are expressed fully and separately, not blended. The example given is like a cow with patches of brown fur and patches of white fur, if it inherited alleles for both brown and white.
So spotted, not light brown, but both brown and white spots.
Exactly. Both traits show up side by side.
Gotcha. And there's a big exception to those homologous pairs too, right? The sex chromosomes.
Ah, yes, the 23rd pair. In humans, females typically have 2 X chromosomes, XX. Males typically have 1 X and one much smaller Y chromosome, XY.
So they're not homologous in males, the X and Y.
Not fully. The Y chromosome is much smaller and is missing many of the genes that are found on the X chromosome. These genes located only on the X are called X-linked genes.
And this matters for inheritance.
It does. This is why certain recessive traits carried on the X chromosome, like red-green color blindness or hemophilia, are much more common in males.
Why is that?
Because males only have 1 X chromosome. If that single X carries the recessive allele for an X-linked trait, they will express the trait because there's no second X chromosome carrying a potentially dominant allele to mask it.
Whereas females have 2 X's.
Females have 2 X's. So for a female to express a recessive X-linked trait, she'd need to inherit the recessive allele on both of her X chromosomes, which is much less likely. She can be a carrier, having one recessive allele masked by a dominant one without showing the trait herself.
That makes sense. Okay, so we've covered how traits are coded in DNA, how alleles work, different inheritance patterns, but how does that DNA information actually get passed on? The new cells when we grow or to the next generation?
Cell division, that's the key.
Okay.
And the source outlines the 2 main types of cell division. First, there's mitosis.
Mitosis, what's that for?
Mitosis is for growth, repair, and replacing old cells in your body. Basically making identical copies of existing body cells, somatic cells.
So making more skin cells or liver cells.
Exactly. A starting cell, which is diploid, meaning it has 2 sets of chromosomes.
1 set from mom, 1 from dad. 46 total in humans.
Right. It first copies all its DNA, so briefly it does double the amount. Then it divides once, splitting those duplicated chromosomes evenly. The result is 2 daughter cells that are genetically identical to the original parent cell and also diploid.
1 diploid cell becomes 2 identical diploid cells. Mitosis. Okay, what's the other type?
The other type is meiosis. Meiosis.
And meiosis. And this one is different.
Very different. Meiosis is specifically for producing gametes, sperm cells in males, egg cells in females. The cells needed for sexual reproduction.
Okay, so not for making more body cells.
No. And the goal here is different too. Meiosis aims to create genetic variation and to produce cells that are haploid.
Haploid.
Meaning they have only 1 set of chromosomes, just 23 total in humans, not 46.
Why only 1 set?
Think about fertilization. If a sperm with 23 chromosomes fuses with an egg with 23 chromosomes, the resulting zygote, the first cell of the new organism, will have
46, the correct diploid number.
Exactly. If gametes were diploid, the chromosome number would double every generation. So meiosis has to have the number.
Okay, how does it do that and create variation?
It's a bit more complex than mitosis. A diploid cell still starts by replicating its DNA, but then it undergoes 2 rounds of division, not just 1.
2 divisions.
Yes. And crucially, during the first division, something amazing happens. The homologous chromosomes pair up very closely.
The one from mom pairs with the one from dad for each chromosome type.
Right, and while they're paired up, they can actually swap segments of DNA between them. This process is called crossing over.
They exchange pieces.
They literally exchange corresponding pieces of genetic material. This shuffles the combination of alleles on each chromosome. It's a major source of genetic variation.
Wow. So the chromosomes that get passed on aren't exactly the same ones the parent inherited.
Nope, they're mosaics of the parent's two chromosomes. After crossing over, the cell divides, meiosis the first, separating the homologous pairs. Then there's a second division, meiosis the second, which is more like mitosis, separating the duplicated strands, sister chromatids.
And the final result?
The final result of meiosis is 4 cells, each of which is haploid, and importantly, each of which is genetically different from the others and from the original parent cell, thanks to crossing over and the random way chromosomes sort into the cells.
4 genetically different haploid gametes. That's meiosis.
That's meiosis, essential for sexual reproduction and diversity.
It's incredible how precisely regulated all that must be. And the actual cell division part, mitosis or meiosis, that's just one phase, right? The M phase.
That's right. The source mentions the cell cycle. Cell division itself is actually a relatively short part of a cell's life.
What's it doing the rest of the time?
Most of the cell's time is spent in interphase. That's the period of growth, carrying out its normal metabolic functions, doing its job. And crucially, if it's going to divide, replicating its DNA during a specific part of interphase called the S phase. Synthesis phase.
Interphase is the living and preparing phase. M phase is the division phase.
Basically, yeah. And this whole cycle is under incredibly tight control.
Controls, like quality control.
Exactly. There are checkpoints within the cell cycle, like security gates. These are points where the cell pauses and checks if everything is okay before proceeding.
What kind of things does it check?
Is the DNA fully replicated?
Yeah.
Is any DNA damaged? Are there enough resources to divide? Are the chromosomes properly attached to the spindle fibers before they separate? These checkpoints are regulated by specific proteins, like p53 and cyclins, mentioned in the source.
And if something's wrong?
If problems detected, the cycle should pause to allow for repair. If the damage is too severe and can't be fixed, the cell should ideally trigger programmed cell death, self-destruction. This process is called apoptosis.
Apoptosis, like a self-destruct button to prevent faulty cells from multiplying.
Precisely. It's a vital safety mechanism.
So, what happens when those crucial checkpoints fail? Or apoptosis doesn't kick in when it should?
That's when things can go very wrong. That's often how you get cancer. Cancer is essentially characterized by cells that ignore the normal signals controlling the cell cycle. They start replicating out of control, dividing when they shouldn't, and they often lose their normal specialized function. They just grow and divide, forming tumors and potentially spreading.
So failed checkpoints are a root cause of cancer.
A major factor, yes. And very often, the reason those control mechanisms fail, or the reason the cell becomes cancerous in the first place, is due to a mutation.
Mutations again, we mentioned those.
Right. A mutation is simply a change in the DNA base sequence. It can be tiny. A single base might get swapped for another during replication, a point mutation, or base might be accidentally inserted or deleted, frameshift mutations.
And even a small change can matter.
Oh, definitely. If that change happens within a gene, it can alter the mRNA sequence, which can then change the amino acid sequence during translation, potentially resulting in a faulty or non-functional protein, like a typo in the recipe changing the final dish.
Okay. And there are bigger mutations, too.
Yes, there can be larger chromosomal mutations, where entire segments of a chromosome are duplicated, deleted, inverted, or moved to another chromosome. The source gives the clear example of Down syndrome.
Right, Down syndrome.
That's typically caused by having an extra full or partial copy of chromosome 21. So instead of 2 copies, cells have 3. Trisomy 21. That extra genetic material disrupts normal development.
So mutations, they sound pretty bad. Cancer, genetic disorders.
They certainly can be bad or even lethal. But the source makes a really great point. Mutations aren't always negative.
They're not.
Nope. Many mutations are actually neutral. They happen in non-coding regions of DNA, or they change a codon that still codes for the same amino acid, so they have no effect. And some mutations can even be beneficial.
Beneficial?
This is where evolution comes in. Think about the grasshopper example from the source. Imagine a population of mostly yellow grasshoppers living in green grass.
Okay. They stand out. Easy prey.
Right. Now imagine a random mutation occurs in one grasshopper's DNA that changes its color to green.
Ah, now it blends in.
Exactly. The green grasshopper is much harder for predators to spot compared to the yellow ones, is more likely to survive
And have babies.
and have babies, passing on that beneficial green color mutation.
Yeah.
This is where the concept of fitness comes in. In biology, fitness isn't about being strong. It's about the ability to survive and reproduce successfully in a particular environment.
So the green mutation increases the grasshopper's fitness in that environment.
Correct. Over generations, the green grasshoppers will leave more offspring than the yellow ones. The frequency of the green allele increases in the population. This process, where traits that enhance survival and reproduction become more common over time, is natural selection.
Natural selection, survival of the fittest, sort of.
Well, fittest, meaning best suited to the current environment to reproduce. As the source says, the individuals with advantageous traits survive and have the most offspring, passing down the trait. Natural selection acting on random mutations is the core driving force behind evolution, the change in populations over long periods.
It's a powerful mechanism. But it has a dark side too, right? Like antibiotic resistance.
Absolutely. That's a perfect and worrying example of natural selection in action now. Bacteria reproduce incredibly quickly, and mutations are always happening randomly.
So sometimes a mutation makes a bacterium resistant to an antibiotic.
Exactly. Maybe a mutation changes the shape of the protein the antibiotic targets so the drug can't bind anymore. When we use that antibiotic, it kills off all the susceptible bacteria,
Leaving the resistant one alive.
leaving the resistant one to thrive and multiply without competition. It passes on its resistance gene, and soon you have a whole population of resistant bacteria. The huge challenge for medicine, driven by the same principles as the grasshopper example.
Yikes. And while we're talking bacteria, the source makes a really important clarification. Bacteria versus viruses. People mix them up.
Constantly. It's a key distinction. Bacteria, remember, are living prokaryotic cells.
Single cell, DNA floating around, reproduce on their own.
Right. They have their own metabolism, they grow. We treat bacterial infections like strep throat or tetanus with antibiotics, which target specific bacterial processes. And remember, many bacteria are beneficial, like the ones in your gut helping digestion. That's symbiosis.
Okay, bacteria are living cells. What about viruses?
Viruses are different, very different. The source points out they aren't even technically cells. Whether they're truly alive is actually debated by scientists.
All right, what are they then?
They're basically just genetic material, DNA or RNA, enclosed in a protein coat, sometimes with an outer envelope. They have no metabolism of their own. They don't grow. They can only reproduce by infecting a host cell and hijacking its cellular machinery to make more copies of themselves.
So they need us or other cells to multiply.
Absolutely. They're like molecular pirates. And crucially, because they lack their own cellular machinery and structure, antibiotics have zero effect on viruses.
Right, antibiotics for bacteria, not viruses.
Correct. Your immune system is what primarily fights off viral infections like the common cold, the flu, or COVID-19. Antiviral drugs exist, but they work very differently from antibiotics.
Good distinction to keep clear. Okay, stepping back from the microscopic. It's just amazing how all these tiny parts, molecules, cells, genes, enzymes come together to build something as complex as a whole organism, your body.
Yeah, trillions of cells cooperating.
And that body is organized into many different organ systems working together.
Exactly. Digestive system, circulatory system, respiratory system. The source gives a great overview of the nervous system as an example of one of these complex systems built from fundamental units.
The nervous system, brain, spinal cord, nerves.
Yep. And the basic functional units are the nerve cells, the neurons. These are highly specialized cells designed to transmit information in the form of electrical signals.
Electrical signals in our bodies.
Absolutely. These signals travel along a long projection of the neuron called the axon. Everything you experience, seeing something, thinking a thought, feeling heat or cold, is ultimately represented by patterns of these electrical signals traveling through networks of neurons to and within your brain.
So my thoughts are electricity.
In a way, yes. The flow of electrical charge along neurons and the chemical communication between them. It's electrochemical signaling.
Wow. And what's fascinating about these signals, these action potentials as the source calls them, it says they're all or nothing. What does that mean?
It means that once a neuron is stimulated enough to fire an action potential, the signal it sends down its axon always has the same strength, the same amplitude, and travels at the same speed for that particular neuron.
So it can't send a weak signal or a strong signal. It's either on or off it.
Exactly. It fires or it doesn't. The difference between feeling, say, a gentle touch versus intense pain isn't the strength of the individual action potentials.
So what is the difference then? How do we feel different intensities?
It comes down to two main things. The frequency of firing how many action potentials are sent per second. More frequent signals can mean a stronger stimulus. And which specific neurons are firing and where those signals are going in the brain. Different patterns encode different information.
Frequency and location pattern. Okay. So how does a neuron actually fire? How does that electrical signal happen?
Let's break it down quickly. A neuron at rest, when it's not firing, maintains an electrical difference across its membrane. It's more negative inside the axon than outside. This is the resting potential, typically around of 70 millivolts.
70 millivolts. Okay, that's the starting point.
Right. Then a stimulus arrives, maybe a chemical signal, a neurotransmitter, from another neuron binds to receptors on the neuron's dendrites, or cell body. This causes ion channels in the membrane to open.
Here, for those charged particles, ions?
Exactly. Positively charged ions, usually sodium, that a plus, start flowing into the neuron, making the inside less negative.
Okay, the voltage changes.
It changes. If the stimulus is strong enough and enough positive ions flow in to raise the membrane potential to a critical threshold level, typically around -55 milliV.
The threshold.
Boom. That triggers the action potential. Voltage-gated sodium channels fly open all at once in that area, and sodium ions rush into the cell, causing a rapid and massive reversal of the membrane potential. The inside becomes positive relative to the outside. This is depolarization.
So it flips from negative to positive inside very quickly.
Very quickly. And this sudden change in voltage triggers the same process in the adjacent patch of the axon membrane, opening its voltage-gated channels. This creates a wave of depolarization, the electrical signal, that travels or propagates all the way down the axon like a row of falling dominoes.
A self-propagating electrical wave.
Cool.
And some neurons send these signals really fast.
Incredibly fast, yes. Some neurons have their axons wrapped in a special fatty insulating layer called a myelin sheath.
Myelin, I've heard of that.
This sheath is actually made of other specialized cells, like Schwann cells in the peripheral nervous system. But the myelin doesn't cover the axon continuously. There are small gaps between the segments of myelin. These gaps are called the nodes of Ranvier.
Nodes of Ranvier. Okay, gaps in the insulation.
Right. In a myelinated axon, the ion flow and depolarization only happen at these nodes. The electrical current effectively jumps from one node to the next, skipping over the insulated parts. This process is called saltatory conduction.
Jumping conduction.
And it's much, much faster than the continuous conduction along an unmyelinated axon. Myelin allows for rapid transmission of nerve impulses over long distances.
Makes sense for quick reflexes and things. Okay, so the signal zips down the axon. What happens when it reaches the very end? How does it talk to the next neuron?
It reaches the junction between two neurons, which is called the synapse.
The synapse.
Typically, it's a tiny gap between the end of the first neuron's axon, the axon terminal or terminal button, and the dendrites or cell body of the next neuron.
A physical gap. The electricity doesn't just jump across.
Generally, no. The electrical signal needs to be converted into a chemical signal to cross that gap, the synaptic cleft.
Okay.
When the action potential arrives at the axon terminal, it triggers the release of chemical messengers called neurotransmitters, which are stored in little sacs called vesicles.
Neurotransmitters. Like dopamine or serotonin?
Exactly. Those are famous examples. These neurotransmitter molecules are released into the synaptic gap. They then diffuse across that tiny space and bind to specific receptor proteins on the membrane of the next neuron, the postsynaptic neuron.
Okay, chemical message sent and received.
Right, and depending on the specific neurotransmitter and the specific receptor it binds to, this binding can have one of two effects on the next neuron. It can either excite it, making it more likely to reach its threshold and fire its own action potential,
passing the signal on,
or it can inhibit it, making it less likely to fire. It's this complex interplay of excitatory and inhibitory signals across trillions of synapses that underlies all brain function, from simple reflexes to complex thought.
Excite or inhibit? Wow, we've really covered a huge range here today.
We really have.
Starting from the very bedrock, the chemistry of the key molecules making up life.
Carbs, lipids, proteins, nucleic acids.
Then zooming into the incredible world of the cell, the fundamental unit, understanding how it powers itself with ATP,
right,
how it controls its internal environment with that membrane.
Homeostasis.
Then diving deep into DNA, the ultimate information storage. how that info is transcribed to RNA and translated into
The
proteins,
central dogma.
how it's passed down through generations via mitosis and meiosis with all the variation from crossing over.
And mutations.
And mutations, how those changes can happen, sometimes causing problems like cancer, but also driving evolution through natural selection.
Right, the grasshoppers.
And finally, looking at how these individual units, like neurons, wire together to form these amazingly complex systems, like our nervous system, all communicating through these tiny electrical pulses and chemical signals across synapses.
It's quite the biological tapestry.
It really is.
And I think what's truly astonishing, and what this source material really helps highlight, is that all this incredible complexity and nonstop activity, the enzymes catalyzing reactions right now, the membranes maintaining balance, the constant production of ATP energy in your mitochondria, the precise choreography of reading your genes and making proteins, cells dividing, neurons firing those action potentials, neurotransmitters being released. Every single bit of it is happening inside you right now. As you listen, it's the absolute foundation of everything you are, everything you do, everything you experience. Understanding these fundamentals, even just scratching the surface like we did, it really does give you a completely new perspective on what it actually means to be alive.
It absolutely does. It's humbling, really. And thinking about that journey we've taken today, from the raw chemistry on a basically hostile early Earth.
From rocks and water.
To the intricate, unbelievably complex processes happening inside our own bodies that allow for thought, for consciousness. We've seen how life emerges from fundamentally chemical principles, right?
Chemistry in disguise.
And how these incredibly complex biological systems, like our brains, arise from simpler unit cells, neurons communicating through these chemical and electrical signals. So maybe here's a thought to leave you with.
Okay.
Considering that your very thoughts, your awareness, your subjective experience of the world right now, it all seems to be the result of these tiny electrical signals racing through your neurons, these chemicals crossing synapses, this intricate dance of molecules and cells we've just spent time exploring. What is that intimate relationship between the physical, chemical, biological reality and our inner conscious world? What does that really tell us about the nature of existence itself?
Deep question to ponder.
Definitely something to think about.